Every living cell contains a complete set of instructions on how to synthesize all of the proteins needed to support life. These instructions are carried in the order of bases along strands of DNA. The process by which cells use this information to synthesize proteins occurs over several steps. The first of these steps is referred to as transcription. During transcription, the cellular machinery targets a short stretch of DNA containing just the information needed to synthesize one or more proteins, protein subunits, or other functional RNAs. The result of transcription is a single-stranded RNA molecule that contains only that information. Cells use transcription to synthesize all RNA, including transfer RNA, ribosomal RNA, and messenger RNA. In order for transcription to occur, a length of the double-stranded DNA needs to be unwound, allowing access to the stretch of bases that encode for the protein to be synthesized. Only one strand of the double strand of DNA is used to synthesize the RNA. This strand is called the template strand. It is also called the non-coding strand. The strand that is not used is called the coding strand or the non-template strand. It is called the coding strand because after transcription, the order of bases in the RNA strand matches the order in the coding strand, except that it contains RNA nucleotides instead of DNA nucleotides. The lengths of DNA used to synthesize individual proteins are relatively short compared to the overall length of an individual chromosome, but they are typically thousands of bases long. The stretch of DNA transcribed contains a number of regions. The upstream region holds the sequence of bases targeted by the cellular machinery to initiate transcription. The coding region is the middle section that actually contains the information needed to synthesize the protein. This is followed by a downstream region where RNA synthesis stops. The transcription process can be divided broadly into three phases. Initiation is when the enzyme complex required for transcription binds to the promoter upstream of the coding region. Once transcription is initiated, it proceeds through elongation, during which time the bulk of the RNA is synthesized. The process ends with termination in the downstream region. The downstream region contains a stretch of DNA structured in a way that it can destabilize the enzyme's attachment to the template strand, terminating transcription. In prokaryotes, the enzyme responsible for RNA synthesis is RNA polymerase. This is referred to as the core enzyme. The core enzyme is fully capable of synthesizing RNA from a DNA template once it is bound to a DNA strand, but it does not readily bind to DNA without the help of other factors. So, for initiation to occur, a protein called a sigma factor must first bind to the core enzyme. The complex formed when a sigma factor binds to an RNA polymerase is called the holoenzyme. This is the biochemically active form of the polymerase, capable of attaching to the promoter regions of genes and initiating transcription. Transcription initiation involves the holoenzyme binding to a promoter through interactions between the sigma factor and specific sequences in the promoter. Bases in the promoter are identified by their position relative to the transcription start site. The first base transcribed is given the label +1. The base just upstream of the start site is counted as -1, and numbers increase in the negative direction from there. Early studies of promoters identified stretches of bases around -10 and -35 that were found to be conserved in many promoters. More recent work has identified additional conserved bases on either side of the original -10 region and further upstream between -40 and -60. Each of these regions are understood to play a role in the way the holoenzyme engages with the promoter region during transcription initiation. They are referred to as the -10 element, the -35 element, the extended -10 region, and the UP elements. In order for initiation to occur, the holoenzyme must bind to the promoter in the proper orientation and at the precise place required for the enzyme to begin RNA synthesis at the +1 base. This means the position and distance between the conserved regions are important. The sigma factor controls this positioning by binding to the template strand at both the -10 and -35 elements, while the core RNA polymerase itself interacts with the UP elements. In addition to its role in positioning the holoenzyme upstream of the sequence to be transcribed, the sigma factor also interacts with bases in the extended -10 region in order to separate the double-stranded DNA and orient the template strand so the core enzyme has access to the individual bases it needs for RNA synthesis to proceed. The fact that the core enzyme cannot initiate transcription without the aid of a sigma factor gives prokaryotes the ability to use sigma factors to regulate gene expression by controlling the types of sigma factors present in the cell and their relative abundance over time. Additional levels of gene regulation are achieved through variations in the bases in the conserved regions. Some promoter regions have many bases in common with the consensus sequence, while others are not as well conserved. Each variation within the -10, -35, extended -10, and UP elements provide prokaryotes with additional opportunities to regulate gene expression. Once the holoenzyme is bound to the DNA in the proper orientation and the double strand has been separated around the transcription start site, RNA synthesis begins. After the sigma factor has completed its role in initiation, it separates from the holoenzyme and the core enzyme continues the elongation of the RNA strand on its own. The elongation proceeds until the polymerase encounters a stop signal or terminator sequence. Termination occurs in two ways: row-dependent termination and row-independent or intrinsic termination. The difference being that row-dependent termination depends on the involvement of an additional protein called a row protein, while intrinsic termination results from interactions between the RNA polymerase and the RNA strand being synthesized without the assistance of other factors. Both types of termination involve the formation of a hairpin loop by the RNA strand being synthesized. Loop formation results from a string of complementary bases in the RNA strand which allows them to bind to each other, forming a loop at the end of the RNA strand as it's being synthesized. This loop is then thought to interfere with the enzyme in a way that causes the rate of RNA synthesis to slow down and pause. During row-dependent termination, the pause in transcription is thought to give the row protein time to bind to that region and destabilize the connection between the RNA polymerase and the template strand. Termination sequences that depend upon intrinsic termination contain a string of uracil bases just downstream of the hairpin loop. This results in a stretch of relatively weak adenine-uracil base pairings, which appears to be able to destabilize the enzyme complex without the involvement of a row protein. In both cases, the destabilization causes the enzyme to fall off of the DNA template, terminating transcription and releasing the completed single-stranded RNA. If it is messenger RNA, then it is available to act as a template for protein synthesis. If it is any other type of RNA, it is ready to be processed for its functional role in the cell. If you found this video helpful, please consider sharing it and giving it a thumbs up. Feel free to comment with any questions or suggestions, and if you want to keep up with the content here at SciencePrimer, click the subscribe button. Thank you for watching.